Okay, good morning, everyone. So I'm James Finley. I'm an associate professor at USC. For those of you who don't know me, my appointment is in the division of biokinesiology and physical therapy. And in my role, I direct the locomotor control lab here. So my research focuses on the neurocontrol and biomechanics of human locomotion.
Hi everyone, sorry I lost track of time.
As it relates both to fundamental principles of control, so thinking about the factors that dictate, when we choose to move, where and how we move, and then we also investigate these principles in populations with movement impairments, such as people after stroke or people with Parkinson's disease. So today I'm going to be leading an interactive workshop on statistical analysis and data visualization. The content from today is largely taken from a course that I teach in research methods in our division. But many of the topics are typically spread over maybe 4 different lectures or more. So, picking and choosing what was appropriate for today is a little bit challenging, but hopefully you all learned something useful. One of the things that I was doing as I was putting together this material is thinking about, who is this for? We have a pretty diverse group here, particularly across career stages. So from undergraduates through full professors. And so the content today is really focused on, some fundamental aspects of largely statistics and how data visualization can really help us better understand what we're doing once we get to the point where we're doing statistical inference. So if you haven't already, please use the QR code to sign in here. And we're going to start with a little interactive poll to get everybody hopefully engaged here. So there's 2 ways you can access this poll. You can just type this URL into either your laptop computer browser or on your phone, and that should work. So give you 1 second to do that. This will also be shown in a smaller form on the following slide. And the purpose of this initial poll is just to frame our discussion and spend a little bit of time thinking and maybe discussing issues surrounding what we're all really trying to do here. So if you type in that URL, it's going to pose a question, which is, what is our primary goal as scientists? This is a really broad, open-ended question, but I think it's really important to guide today's discussion. You have the opportunity to upvote people's answers. So if you see an answer that you agree with, please give a thumbs up, and we'll see if we can come to any sort of general consensus here. Thanks everybody for chiming in. We already have lots of responses. Sometimes this happens, we do this and we get 0 responses, so this is nice. I'll also state that it's, for those people who are leading these workshops, it's often a little hard to manage. I can't see who all is there. Let's see if I can change this. Well, I see that there are 35 participants, including some watch parties. So we have a nice sample of people here. Okay, so lots of answers here, and I think we are at a point where we can start thinking about consistent themes. So I have, in just these first 4 responses, we have 2 things, right? We have answering questions, generating knowledge. Okay, so that's a pretty common theme across all of these responses. Some people have things related to truth, so that's a very, I guess, deep topic. Oh, going the wrong way here. So, but basically the general idea is that we want to learn things about the world. In learning things, we generate knowledge. And I think that's, a consistent theme across everyone's response. So one of the things I want everybody to keep in mind as we go through today is how can we be confident about the knowledge that we generate? What are some of the assumptions that underlie the methods we use when we generate knowledge? And more importantly, how can we make sense of that knowledge and do what we call generalize it to things outside of our particular research studies? So if you have any comments as we go, feel free to interact. I'll point out, if people haven't seen it, I posted a link in our MC chat with a link to a Google Drive that has some MATLAB code that we'll be working on today. Can you share that link in our lab Slack?
It's asking me with permission.
For the
Create an folder, yeah.
Okay, you can just request, and I think somebody can give you access.
Yeah, I can share access if needed.
Okay, thank you, Victoria. All right, so let's talk a little bit about types of research. So everybody involved in IMSI is doing presumably one of these types of research, and I think it's important to acknowledge that our research may have different goals. So in some cases, we're interested in doing descriptive studies where maybe we're studying a population of people, other organisms, and we want to characterize something that we observe in those participants, if you will. And so there we're not necessarily doing hypothesis testing, but we're interested in what we're going to call descriptive statistics. In other cases, we might be developing methods. So when we're developing, for instance, measurement tools or assessments when we're dealing with animal behavior, one of the things that's really important about these tools is we might call their validity and reliability, right? So how accurate are our tools? And if we were to make the same recording from the same, person, organism in the same context, how consistent are those responses? So answering both of those questions about validity and reliability require one or more types of statistics. Moving sort of maybe in some ways up the hierarchy, we often do studies where we're interested in relationships. So we're measuring multiple variables and we want to know how they relate or co-vary with one another. So there are various statistical tests, regression, correlation that we might use to capture these relationships. And then, one of the sort of big goals of what most of us do is to try to understand cause-effect relationships. So while we can easily establish correlation, it can often be much more difficult to determine cause-effect relationships. So that's really the purpose of experimental research. And if we have time, we can talk about factors that influence our ability to establish those relationships. All right, so there's kind of two challenges that I want to start with to set up the rest of today's workshop. The first is this idea of generalization. So when we talk about generating knowledge, answering questions, one of the things that we often do in science is that we're actually interested in some population. So let me get my laser pointer. Okay. So in our work, if let's say we're working with people post-stroke and we're interested in factors that influence balance control, really what we care about is in the large population of people post-stroke who have certain characteristics, how do they maintain balance? What are the factors that influence that? But in almost all cases, right, we can't study the whole population. So clearly we do studies in the lab where we sample from this population. And so this is the set of people, organisms from which we actually observe behavior. And our assumption oftentimes is that what we observe in this sample is representative of the whole, right? Representative of the population. Okay, so we'll talk about some issues when that is and maybe is not the case. The second challenge is in other cases, we're not necessarily interested in a population, but maybe we're interested in an individual. So you may have, one or more types of organisms that we study depending on your lab. And what we're trying to infer is something about the individual, right? And so, for instance, in our lab, we study decision-making. And one of the things that we would like to know is what are individual differences in how people weigh things like risk and effort during walking. So there we're going to make some set of measurements on an individual level. And again, we have a similar challenge as what was illustrated on the previous slide. Right, so at a high level, we know that if I just measure one measurement on an individual, I don't necessarily think that I can't necessarily infer much about that person's priorities, right? So I need multiple measurements. So this is still a sampling question or an issue at an individual level. So we can think about how much data do I need from an individual so that I can be confident about the knowledge I generate about that, again, person/organism. So, so I mentioned this, that we're often interested in identifying causal relationships, and generally we think that there are 3 different things that we need if we want to know if A causes B. So a lot of what we'll talk about today is related to issues of variability. And so there are types of variability that we might think of. We can consider good, and then those that we want to minimize. So this first idea here, concomitant variation, is basically the good variability. Right? So if I'm manipulating something, so the thing I control, right, we call our independent variable. I measure something, we call that our dependent variable. I hope that there's variation, right, in our dependent variable when I change the thing I'm controlling. So that's the variation that we really like, right? We need that variation if we're going to establish relationships. The second thing that we need to establish causality is this idea of the cause and the effect have to co-occur. We won't talk a lot about this, but basic idea is that if some effect is already present, then I can't establish cause and effect. The last point is one that's more important for today, which is control of other sources of variability. Right? So here we can think about these what are called internal agents or confounding variables as being the variability we don't like. So we either want to minimize those sources of variability or measure things that capture those sources of variability. So this last point is really kind of the core of some of what we'll be discussing today, both with statistics and data visualization. Okay, so when we talk about these sorts of variability that we maybe don't want, we can think about different classes. So there's what's called extraneous variability. So this often has to do with, you can think about variability that you introduce into your data, potentially yourself, right? So if I am making multiple measurements on a set of participants, maybe I'm inconsistent from day to day, right? So that adds variability that is not useful for my statistical analysis. If I have tools that are imprecise and inaccurate, that's going to generate variability that again reduces my ability to detect or determine cause and effect. And then there are lots of different characteristics of our participants that also introduce sources of variability that we may not be interested in. So as an example, right, in our study, when we were interested in decision-making and human behavior, one of the things that is highly variable that we can't always control is the issue of, I'm going to generally call this, alertness. There are different characteristics that might relate to this, and that's misspelled. But the general idea is that, how much are people paying attention and staying on task? Clearly, people's attention waxes and wanes during an experiment, and that's going to contribute to some of our variability. Right. And then lastly, we can think about this idea of confounding variables where these are things that are going to, again, prevent us from determining cause and effect. Okay, so variability is kind of the core of today's discussion. And I like to think about statistics as really being tools that we use to both quantify and explain sources of variability. One thing that I'll say was maybe, as a bit of background, my degrees are in engineering, and I will hand wavily say that engineers are often not very good at statistics. Like I don't feel like I had a very strong training in statistics as an engineer, even though we did a lot of math. And so when we're doing statistics in experimental or in research, we're often, using tools that assume that we have large quantities of data. That's the first part. And we are, like we said earlier, trying to infer something about a population from a sample that we studied. Okay. And so we're going to use two different types of statistics mostly in our work. We're going to use descriptive statistics to summarize our data. And then what most people are interested in is the result of inferential statistics, right? So we do our t-test, our ANOVA, our regression. And one thing that may or may not be clear to everyone here is that when we do inferential statistics, we are doing that under the assumption of some sort of model of our data behavior population. So let's start with thinking about some factors that we want to be careful about when using descriptive statistics. Okay. So I'll breeze through these. So I think most everybody is familiar with basic terms that we use in descriptive statistics. So often we are thinking about, for instance, a set of measurements that we might represent like this. Right? This is probability distribution of our measures, or something like a histogram. We're often interested in the midpoint, this measure of central tendency, which we might illustrate as our mean, and the spread in the data, which we might quantify as our standard deviation. Basic ideas here are that when we report the mean and standard deviation of a set of measurements, we're in essence assuming that those measurements come from a normal distribution. We might also be interested in the relationship between multiple measurements, right? Where if I have two measurements here and I have many values, maybe I want to think about how tightly related or correlated those measures are. And then we've already introduced our measures of dispersion up here. So the one key point I want to just make here is that when we think about measures of dispersion, one term that comes up a lot in the world of statistics is this idea of variance. And so variance, of course, is the essentially the average. You can think about it as the average deviation of all of your data points from the mean of your data set. And the equation for variance is here. We won't spend much time looking at this, but we're going to look at some data sets shortly that have essentially the same statistical properties, but are very different. And one of the key take-home points from today is that looking at your data early and often through your analysis is really critical for making sense of the statistical results that you obtain. So what we're going to do is we're going to start by looking at this set of data referred to as Anscombe's quartet. I don't know if there's not an easy way to poll people. Hopefully, I'm assuming some people have heard of this and maybe many haven't. But these are 4 different data sets that when you see them are clearly very different, but they have the exact same statistical properties. So this is one of my favorite illustrations of how statistics can be essentially misleading. So what we're going to do, we're going to move over to MATLAB now. There is a script in the Google Drive called Anscombe's quartet. And you can basically run that script. And we're going to answer the following questions here. All right, so let's see if I can easily do this. So I need to go here. Okay, can you all see my MATLAB window now?
Yeah.
Okay, great. Okay, where are we? Okay, so in the files that you downloaded, there's a CSV file called Enscomb's Quartet. So what we're going to do is we're going to read the data from that CSV file into MATLAB. We're going to create a variable just called data. And essentially what this script is going to do is just plot the data. So for those of you who are maybe new to MATLAB programming, we're going to store some information, some statistical information about these data sets. So we're going to generate that statistical information in a loop, starting here at line 12. So I'm starting by doing what's called pre-allocation, where I set aside space in MATLAB to store the outcomes of our essentially statistical analysis. We're going to generate a figure, and then we'll take a look at the data first. So you can go ahead and press run. Should work. Make sure that your CSV file is stored in the same folder as this script. So hopefully that's working for people. Does that work for people here? Yes, okay. So here's what's generated when you run this script. So first what we're going to do is briefly look at the data matrix and then the figure. So if you go to your MATLAB workspace, so for those, again, who are new to MATLAB, your MATLAB interface probably looks a little bit different from mine. We're interested right now in the workspace. So here's where we can see all the variables that are that we've generated. And so the variable data has 11 rows and 8 columns. So I can easily visualize that here. And the way these data are organized is that they're organized in pairs of columns. So the first 2 columns are, the first is, we would call this X1, Y1, then columns 3 and 4 would be X2, Y2, and so on. So the figure is going to plot the data from the first 2 pairs as data set 1, the second pair as data set 2, and so on. So let's take a look at this figure. So for each of the data sets, we're plotting the X values along the horizontal axis, the Y values along the vertical axis. And so it's pretty straightforward. I think if we were to visualize these data sets, everybody would say that these data sets are quite different. And what we're doing, the line there is a linear least squares fit to the data. And again, intuitively, we would say, hey, that line really is only appropriate in this first data set, right? In all these other data sets, there are issues with the data that make it such that fitting a line really doesn't make any sense. So now let's imagine that some group has published this data and you don't have this figure, but instead all you have are the summary statistics from each of the 4 data sets. So the first question is, well, let's get some descriptive statistics. So what we're going to do is compute the average or the mean of the X and Y values for all 4 of these data sets. So we can go to our command window. If I just type mean of data, what this is going to do is compute the mean across every column in this data set. So we know the data look different. Let's see if the means are different. So again, the data are pairwise. So we have X1, Y1, so 9 and 7.5, X2, Y2, 9 and 7.5, and so on. So even though the data look very different, the means of all the data sets are basically exactly the same. Now let's do the same thing with the standard deviation. I'll try not to do shortcuts here. So if I type STD of data, now I get the standard deviation of each column, and I see the same pattern, right? All the data, each data set has the same standard deviation for the X and Y values. Okay, so that seems curious if I just look at the data set. If we were to place a bet ahead of time, most, and you hadn't seen this data before, you wouldn't assume that these statistics are the same. And so finally, what we're going to do is look at our regression statistics. So when we fit a line to our data, what we're going to do is there are several parameters that we're going to obtain. We're going to get a set of coefficients, we might call them betas, that characterize the y-intercept of the data set and the slope of this line. We're also going to get a measure of r squared, which is the variance in in y that's explained by x. And we typically think about this as the goodness of fit. So again, intuitively, if we look at these data sets, we would say that this line does not fit the data well for data set 2. It doesn't seem to fit the data well for data set 4, right? So we might expect r square values that differ across these data sets. So in our simulation code, or analysis code, if we scroll down to our for loop, we use a function called regress in MATLAB to do a linear regression for each data set, we're regressing y onto x. And we have a variable b that stores our slope and intercept for each of those regression models. And then we have a variable stats, where the first column in stats holds all of our r squared values. So you can probably see where this is going. If I look at my model parameters in V, so the first is my intercept, and the second parameter is let me make sure I'm doing this. The first parameter is my slope, the second is my intercept. So slope on row 1, intercept on row 2. Each column is a different data set. So again, here I see that my regression model fits are the exact same for all 4 of those data sets. And lastly, if I check my r square values, which are in the first column of the stats matrix, I see that my r square values are basically all the same. Okay. All right, so this is something that I think for anybody who does statistics, you should just burn this into your memory. Looking at summary statistics alone, whether they are descriptive, right, mean and standard deviation, or whether they are inferential, model parameters, r square values, they don't necessarily tell you much about how the underlying data look. All right, so the sort of intermediate take-home points here are, essentially rewording what I just said. Descriptive statistics may not fully capture the key characteristics of your data. This is particularly true when we have small samples, which is pretty common for a lot of the work we do in, whether it's biomechanics, human models, animal models. We have to be very careful about what we're inferring. And one of the key tools that you have to better understand your data is, of course, visualizing your data. So I'm a firm believer that you should visualize your data at every step from the time you collect it through each step in your analysis pipeline, as you lose information along the way. All right, I'll just pause here for a minute in case people have any questions, comments, thoughts. And again, I can't see anybody.
So you have
Yep.
a question. So for the variability of my data in this way, it would be the variance a way to characterize the variability of my data better? Yeah,
so the question is what are ways to characterize the variability in your data? Mm-hmm. So we can compute our measures of, whether it's the range or the standard deviation, right? These are quantitative measures of variation. But I think that you want to do these in addition to visualizing the data, right? Because if you compute the standard deviation, but you have outliers, right, it's going to be sensitive to those outliers.
Okay.
Any other questions?
Oh, we have a question here in the chat from Hansel. Do you have comments on different definitions of R squared?
I do not. So I think this might, relate to things related to the, variance explained versus the, what is it? The, I forget the other term off the top of my head, but there are different, essentially, sort of ways you can compute R squared and some slightly different interpretations. That's kind of beyond, I think, what I was at least prepared for discussing today. Hansel, do you have any comments?
Not really. I'm just confused about it all the time. Most of the times we do correlation coefficient and get the squared of that value and call it R squared. But for some fields, it seems like they do 1 minus something, which can sometimes give you value less than 0. So
Yeah. Okay, so okay, I can at least sort of sketch what you're saying here. So one idea is that you have your data, you compute the correlation coefficient, right? So R is equal to 0.8, and then what you're saying is that you can say R squared is going to be 0.64, right? You just square R. Another way that people will compute this is 1 minus the variance of your residuals, I think, over the variance in your original data, something like this. And so sometimes you can fit, if your model is really poor, the numerator can be bigger than the denominator, and you can get negative values here. So I don't actually have a good, frankly, good understanding of when one is more appropriate than the other. I'll try to find something and share it on our IMSI Slack. And if anybody else has an answer to that question, feel free to chime in. I'm not a statistician. All right.
Lisa has her hand up.
Yeah, I think I was just going to quickly say that, I think like goodness of fit tests is an area where there's currently been a lot of controversy. And I know that I've had lengthy discussions with Madhu Venkadesan about this recently. And you can find a lot of papers online talking about, why R squared is positively misleading. And and I think most of the examples are coming from like economics and predicting like the price of, pork bellies in, 6 months or something like that, or, anyway, the point being, there are typically models that have like a lot of independent variables that are trying to be used to predict like a single like output variable. But the point being, I think, figuring out whether R squared is the appropriate goodness of fit measurement for your data is another whole can of worms.
Okay, so this is related to an important point that I didn't, plan to discuss. So there are two terms that we think about when we're doing regression. So when we fit a model to our data, right, and we get an R squared, what we're really doing, we're explaining variance in our measurements. Now, the problem is that sometimes we use the terminology of our independent variables as predictors. So in sort of formal statistics, machine learning, pick your field, that's actually we're not doing prediction. So, and with prediction, what we might do is start with a model, and given some values of our independent variable, we then compute from that model what the dependent variable should be, and we compare that with reality. So to Kisa's point, the more parameters you add in a model, the better your explanation, but you can also make your prediction worse. So that's a thing you always have to be careful about in statistical modeling. Okay, so yeah, so that's a general take-home point. I think there's three scripts and 3 major take-home points for today. All right, so what we learned from Anscombe's quartet is that really the only reason why we know that those descriptive statistics are misleading is because we visualized our data, right? If you didn't do that, you would have no idea. And so what do we think about when we actually do data visualization? So this is going to be a rapid overview of a paper that I really like. So for those you may or may not be familiar with, so PLOS Computational Biology is a journal that has a series called 10 Simple Rules. These are really nice papers, kind of perspectives that different authors, groups will write. And this will give you 10 tips for all sorts of things. It could be, here we're talking data visualization. You can find papers on how to run your lab, how to make usable software and research, all sorts of things. So highly recommend this series. And so one of the things that I always like to do, this could actually be well, I won't say that. Sometimes I think it's my alternate career would just be data visualization because I think it's a lot of fun. This is an example of a figure that you might see in something like the New York Times, right? Which is illustrating causes of cancer death among men and women using color, text, shading to illustrate many features of the data set. Now, one of the things I used to think when I saw figures like this is that, there was some illustrator who used some data visualization program to really make this look very clean. And the reality is that you can create this exact figure in MATLAB using only code. So major take-home point for this next section is when you generate figures, particularly if you're going to share them with anybody else, take the time to make sure that your figures are conveying the key ideas that you'd like them to. So what are some steps for this process? So the main first thing is don't trust the defaults. So regardless of what program you're using, the default settings are going to be bad. So there are different features of your data, your figures that you can control. And this is my personal opinion about MATLAB figures. The font's often too small, lines are too thin. And so what we're going to do is try to get comfortable specifying the settings of our figure using code. So we're going to move back to MATLAB and we're going to run this script called show the data. So let's go back to our MATLAB editor. All right, so we're going to use the exact same data set because simple. And we're going to do a couple of things here. So first, we're going to I'm going to reiterate the first point I made with this data set in a slightly different way. So we're going to create a figure that has 2 panels. And the first panel is going to be a bar plot of the Y values in each data set. And so we might call this an error bar plot. We're going to have some error bars. And then the second figure or panel is going to be what's called a dot plot, where we show the medians of each data set. So there's a lot of code that's commented out because we're going to come back to that. So now, if I just create a bar plot with error bars and a dot plot in MATLAB, so if you just press run, this is going to generate a figure with all of the MATLAB defaults. And for me, this is what it looks like. This is horrible, right? This is this is like a hideous figure. You would hopefully not see this in most of the papers that you read. But if you don't do anything else, this is the baseline. When I was a grad student, what I would do is take a figure like this, I would export it to Illustrator, I would resize the axes, I would change all the colors, I'd add some labels. It was very tedious. I think a lot of people still do that. And that was because at the time I had no idea how to modify these things. I mean, what I'll say is, programmatically, right? Writing code to set everything that I want. So, yeah, so the take-home point of this figure, right, is again, if I take that same data set, I do a bar plot with error bars, it looks like all of my data sets are the same. But if I do a dot plot, right, I can see that the data are spread in very different ways. Some data sets have outliers and so on. So what are some of the principles that we want to consider when we're making figures? So I'm going to go back to our PowerPoint here. See how we're doing on time. So 30. I think we're right on time. All right, so these are 8 steps that this is just what this is not gospel. These are just 8 steps that I use, and you can replace MATLAB with, R or Python or whatever your tool of choice is. But one of the things that I think is always important when you start making figures is having systematic process. So we'll talk about each of these 8 steps, and hopefully everybody learns at least one thing that might be useful. So the first thing is, of course, you always want to determine why you're making a figure. So people in my lab will know, so I use Slack a lot in the lab. One of my favorite things is if people send me data, I almost always just stop what I'm doing and I look at the data because it's really exciting. But what I don't like is if somebody just says, here's the data, right? And so what you always want to do when you're sharing data is to say, what was the goal, right? You share your data, and what did you interpret? So, so you always want to think about before I make a figure, what is the point? What am I trying to communicate to my audience? And this can be any number of the things that we've discussed already: descriptive statistics, associations, inferential statistics. The way that I think about data visualization is that we want to make a figure when an illustration is a better way to convey a piece of information than text. Okay, so sometimes text is sufficient, other times you need to visually convey. I like to do a lot of sketching, so I like to think about how I expect the data to look before I make a figure. So these are just some hand-drawn sketches where I can think about different ways to illustrate a set of data. So at the top, I have a histogram with a probability density or distribution. The bottom 3 examples, I think these are most relevant for right now. Right, so in the figure we just generated in MATLAB, we can have the same data set where we can visualize it as a bar plot with error bars or a dot plot. And I think people's intuitive understanding of the data differs, wildly based on how the variability of the data is illustrated. So then once you have an idea of what you would like to show, you then move on to selecting an appropriate function. So one thing I like to tell people is that if you can think about a way to visualize data, again, I historically primarily use MATLAB, but the same is true for most other languages. There's probably a way to create that figure in MATLAB. So here's just a subset of functions that you might use for data visualization in MATLAB. I highly encourage people to look at the MATLAB help for all these functions so that you know what kinds of characteristics or features you can change. And then finally, MATLAB has what's called the file exchange. And this is a place where people will create custom bits of code for everything from data visualization to machine learning. And they'll share that code with other users. And so you can often find useful functions there or useful scripts that someone else has created. If you yourself create scripts that you think might be useful for other people, I highly recommend sharing them on the file exchange or other places. All right, so now you know what function you want to use. The next thing is, of course, to create your figure. The key thing that happens, I think, when most people start making figures is you use, plot or scatter or whatever, and then you show someone the data. But hopefully most of you have already had this drilled into your heads, but you always want to add informative axis labels, ideally, using full words and units so that people know what you're showing on each value, each axis, and they can interpret the numerical values. You can even do things in MATLAB like add text to your figures. I've written code, like if I want to compare, it shows statistical significance. You can just write code that draws lines to do your little statistical significance indication and add your asterisk or whatever character you'd like. So some basic functions that we're going to use are labeling our axes, maybe adding legends. Text is the one that you use to generate text on your figure. So once you generate your figure and the data are illustrated appropriately and you have your things labeled, next is the part where you can start to get a lot more control. So here's some things that I will often do. Again, when I was a PhD student, I would generate figures in MATLAB, often use the default or sort of like manually adjust things. And I would export it, manipulate it in Illustrator, and then submit it to the journal. And there were a couple of times where once the proofs came back for my paper, often the figure was, unreasonably large, right? It's like, why is this figure so big on this journal page? This looks silly. And it's because the actual image file that I submitted was large. I didn't have a good sense of how to scale my illustrations. So one of the things that I like to do in MATLAB, and I'll show you how to do this in code shortly, is decide, okay, if I want to publish this figure, do I want it to be 1 column wide, 1.5 columns, or 2 columns in a typical 2-column journal format? So you can get these dimensions from many different journals. But once you know how large you want your figure to be in print, you can set those parameters in your code. And so, I'll do things like specify font sizes, marker types, all sorts of things that I used to do manually in Illustrator, and just put it in the code. One of the reasons why this is really helpful is because we often will work hard on data analysis, making a figure, submitting it to a journal. But then if my reviewers ask me to redo some of the analysis and generate a new figure, I don't want to go through the process of manually having to add these features in a separate program. All right, so then finally, what we're going to do is export and save our figure. So of course, give it some informative name, not fig 1, fig 2. And I will often export multiple versions of the figure. So I'll save this sort of MATLAB version in case I want to edit it later in MATLAB. I'll export a PDF, both because if you need to annotate it in another program, PDFs are easy to work with. And then sometimes I just use the PDF, sort of screen capture the image in some way and export that for a presentation. And then finally, some journals require image-specific files. So TIFF files are one of the highest resolution image types that you can use for journals. All right, so now let's work through an example. So we're going to go back to our script to show the data in MATLAB. Click over here. So now we're going to uncover some of these features. So on lines 11 through 17, this is an example of me specifying the features of our bar plot. So one thing that you may or may not have noticed, so when I use each of these plot functions, I'm actually assigning a variable, assigning the plot to a variable. So I have E for my error bar, B for bar. The reason I do that is because then I can use the MATLAB function set to specify the parameters of those figures. So for my bar graph, the bar has both what we call a face color, that's the main color, as well as an edge color, which is the outside, line width, many different properties. Same thing for the error bar. I can specify some of those properties. I then specify the X and Y limits, add some labels, specify my X labels, remove the box from the outside of the figure. So what I'm going to do is highlight lines 11 through 17, and I'm just going to uncomment those. So I can right click and uncomment, or I can do Control-Shift-R or the comparable thing on a Mac. So again, I just remove the commenting from lines 11 through 17. So just as a reminder, this is our original figure. So this is the one we're going to try to make what I'm going to call publication ready. So for our dot plot, by default, right, our dots are, I think they're different colors, or the lines are different color. No, the lines for my medians are different colors. It's hard to see those lines. The range of my axes is very different from figure 1 to panel 2. So let's try to make this a little nicer. So now I'm going to uncomment lines 25 through 29. So again, for my dot plot, now I like to specify the line width for each of those points. I like to have black outline with a gray face. I think it looks a little better. And then I'm going to specify for my axis how thick the lines are for the axis, and add some labels. So finally, what I'm going to do is set the figure dimensions for publication. So again, these are the numbers that I often will use. So I've decided that I want this figure, because it has 2 panels, I want it to be 1.5 columns in the paper. So I'll have in the paper, the figure is 1.5 columns, and maybe my legend is to the right of the figure. So I'm going to uncomment lines 33 through 38. So here's where I specify my width. So this is in centimeters. I then use set again. So now when I use set, instead of feeding it the name of my figure, I had S up here, I had E and B. I use something called GCF. And this stands for get current figure. So it says, okay, whatever figure is open, set its units to centimeters. And then the position property of this figure is where I'm going to specify the size of the figure. And then finally, I'm going to save it as a FIG file and a PDF. So now you should be able to run this code again. And we can compare the before and after. So here's our before figure 2 for me. Here's our after figure 3. So now I have nice labels on my x-axis for each of my groups. The font size seems appropriate for the scale of the figure. I've cleaned up my bar graphs, made things grayscale, and changed the dimensions. So to get a better intuitive understanding of what it means to set the dimensions of the figure, if you go to your MATLAB command window, if you go to your current folder, hopefully you have this current folder sub-window visible. If not, it's okay. But we're going to open the PDF that we created. So I think I saved it as, yes, show the data PDF. So for me in Windows, I can just right click this file and choose open outside of MATLAB, and that's going to use Acrobat to open the figure. So it's opening now. And so now when I look at my figure, get my windows working properly. Oops. So now in Illustrator, right, I can see that this is, you know, an 8.5 by 11 sheet of paper. And my figure, you know, if this were in a journal, would be about 1.5 columns. And it looks, you know, clean and appropriately sized. Okay, so that wraps up our sort of second part of today's workshop. So any questions, comments, things that you know, things that you like to do when you generate figures? A minute.
So saving in JPG would lose like quality of the image?
So you know, a JPEG is an image. So you know, when you generate an image, a JPEG is what's called a compressed image. So it's a smaller file size than a TIFF file. So a TIFF file has more information. These are things that are often most important when you have very detailed images like photos and things. It doesn't matter as much for like line graphs, but yeah, I just have personally choose to save them in the higher resolution.
Do you still move this over to Illustrator or PowerPoint or something to add in like a sub panel, like A and B labels?
Great question. I basically got to the point where I forced myself to try to do everything in MATLAB. And it turns out that you can also add, you know, using the text command in MATLAB, you can add, let me see if I can have my cursor. Let's say I wanted to add B here, right, and A here. You can just use the text command in MATLAB. And so the way that the text command works is that you feed it the X and Y values of where you want your text to be. And so if I want it to be sort of to the top left of this panel, my X value is going to be negative, right? So it's, you know, 0 is here, so it's going to be like -1. My Y value might be 14. Yeah. So, and you can, you know, I've written code that sort of like normalizes the position based on the range of the Y-axis and stuff like that. So I'm basically convinced that most everything we would normally do in Illustrator, you could just write code to do that.
Thanks.
Other questions though? All right, so let's move to the final part of today's workshop. So we're going to go back to statistics. So a lot of what we've been talking about so far is really related to descriptive statistics. So thinking about what we compute, how that relates to what we see, and making sure those things are either consistent or at least that we're making a proper interpretation. So now we're going to talk a little bit about inferential statistics. So what happens when we actually want to do hypothesis testing? Now, again, this could be a whole course, so figuring out exactly what I wanted to share today was a bit of a challenge. So I'm going to focus really on kind of one major issue and place of confusion that at least I had for many, many years. And that's the end game. So what are we actually trying to do? So this is a you know, a figure that illustrates the idea of a data analysis pipeline. So we start with some experimental design, we then collect data, we get raw data, we process it, we fit some statistical model, And then finally, at the end of the day, we get a p-value. And so for a lot of us, you know, whole fields, individual people, we sort of live and die by the p-value. And if p is, we talked about this yesterday, right? If p is 0.051, we like have a breakdown. If p is 0.049, we're, you know, as happy as can be, right? So that's, you know, how a lot of science or people work. And so we'll talk a little bit about like, what are we even doing? Like, what is a p-value? How should we really be thinking about this? And what's maybe an alternative to the norm? So the thing that really got me thinking about this issue more deeply, like a lot of people in science, is this paper that came out in 2015, which is called Estimating the Reproducibility of Psychological Science. This is basically a large group of people in psychology that what they did was essentially they chose a range of studies that were published in prominent psychology journals, and they tried to reproduce the effects. So they worked with the original authors, they got the original study materials, and they just reran the study in a new sample. And they did this for many different studies. So the most striking figure from this paper, right, is that if you look at the p-values for the original studies, so that's on the left here, this horizontal dashed line is 0.05. So unsurprisingly, almost every study that was published had a p-value of less than 0.05. However, in the replications, you see a much wider distribution of p-values. So this white dot is the median p-value. So over half the studies had a p-value of above like 0.2 when in replication, and only a very small percentage of those studies would, what we would say, replicated in the sense that there was a p less than 0.05. So this paper is in the folder that we shared today. Highly recommend everybody read. So if we have time, we can get into sort of what the problem is here. But at a high level, there are several factors that dictate, you know, whether we choose to publish, how stringent we are in evaluating the rigor of our results. And this has major implications for whether our results generalize to the real world. So what we're going to do is, one of my favorite things to do is to, you know, build some intuition about p-values. So first, the definition. Everybody who takes statistics probably gets this definition at some time. So our p-value, which we often call significance level, is a probability. And so, you know, I can read it here. Even just reading it, it never sounds very helpful. Right? The probability of rejecting the null hypothesis when the null hypothesis is true, right? That's really wordy. Like, what are we even talking about here? It's also confusing because people will say, oh, this is related to your false positive rate or your type 1 error. So additional jargon that's really confusing. This is before you even do any statistics. So first we need to think about, okay, what is a null hypothesis? So the fundamental idea when we talk about p-values is that the null hypothesis is true. So here's an example, right? So let's say I'm a sports biomechanist, and I'm interested in sports, in hamstrings injury. Well, maybe I'm studying a set of distance runners or soccer players, and I'm interested in whether the total distance people run each week is associated with the, you know, frequency or severity of hamstring injuries. So my null might be that there's no relationship, right? So the weekly running distance is not associated with hamstring strain. So it's a little counterintuitive because we often just like, we don't, sometimes we don't express a null hypothesis, or we just don't believe, we go into a study believing that the null hypothesis is false. So now what we're going to do is take some time to understand a p-value. So we're going to use a simulation to first create a case where our null hypothesis is true. Then we're going to run a bunch of simulations where we see how often we're going to reject the null hypothesis. So rejecting the null hypothesis, right? When we say p is less than 0.05, for most people, that's sufficient information to reject the null hypothesis. So let's see what happens if we do, many studies where the null hypothesis is true. So let's open this script, understanding p-values. All right, so lots of introduction here. So I'm going to create a sort of synthetic experiment. So let's imagine that we are, again, we're sports biomechanists, and we're doing an experiment where we want to test the effects of this supplement. Right? We want to see how well this supplement increases long jump performance in Olympic male athletes. We have a population, we have an intervention. So what's our magic supplement? It's water. Right? So our supplement is going to be 1 ounce of plain tap water. So we're going to have 2 groups, right? A control group that doesn't take the supplement, an intervention group that drinks 1 ounce of water, and we want to see, if their long jump performance differs. And so this is setting up an idea where, like, in this case, the null hypothesis basically has to be true. There's no way that drinking 1 ounce of water is going to affect your long jump performance. So now let's think about what happens if we run many, many experiments where, let's say 1,000 different labs ran an experiment to test this hypothesis. So the key idea here is that we have a population, right? This is all of our male Olympic long jumpers. We're going to specify some parameters of our population. So in our control population, the average long jump is going to be 8 meters plus or minus 20 centimeters. In our test group that drinks 1 ounce of water, the same thing should be true, right? We know that there's no effect. So this is our population. Now what we're going to do in simulation, we're going to sample from that population, right? This is what we do in an experiment. So I'm just arbitrarily setting the sample size at 100. So we're going to take 100 people from each of these populations. And then we're going to simulate an experiment. So we don't have a lot of time here, but basically if I want to sample from a population and I know the mean and the standard deviation of that population, this is basically all I need to do in simulation. So I am going to generate some random numbers, that set of random numbers, so it has the same number of values as our sample size. I'm going to multiply all those random numbers by the standard deviation of our population, and then I'm going to add the mean. Okay, so this is going to give me a data set that looks like it's sampled from the population. Okay, so I have a control group and a test group. So this is just going to give me the long jump distances for, in this case, 100 control participants and 100 test participants. Then what I'm going to do is run a t-test. So I'm just going to compare the average long jump distance between the control and test group. So now if I know that the null hypothesis is true, then I should, essentially all of my p-values you might expect would be greater than 0.05. So let's see what we get when we do this. Okay, so we're going to first we're going to start with just one experiment. Okay, so let's press run here. So I think a couple figures should pop up, maybe just one. This figure doesn't make a lot of sense right now because we only have one experiment. But what we can do is we can get our p-value for this one experiment. So if I go to my main MATLAB window, I scroll down to p. So p is, for me, is 0.18. So in other words, I run this simulated experiment and I don't reject the null hypothesis. So my experiment is actually consistent with reality, with the population. Now let's do this, instead of just doing one experiment, let's do 1,000 experiments. So on line 25, I'm just going to change the 1 to 1,000. And the key thing that we're going to see is, of these 1,000 experiments, how many of them get p less than 0.05? Does anybody have any guesses as to how many of the experiments will have p less than 0.05? If you do, you can just drop it in the chat. So my hope is that after this, everyone will have a much more intuitive sense of what p-values are. So if I press run, I just ran 1,000 experiments in 40 milliseconds or 200 milliseconds, and I get a distribution of p-values. So I get p-values between 0 and 1, right? It's a probability, so we know that's the range. This distribution is more or less what we would call a uniform. If I did, like 1 million simulations, this would be flat. And so what do we know about a uniform distribution? We know that any value within this interval between 0 and 1 has an equal likelihood of happening. So we can't really see, right? So these bars, this is from 0 to 0.01. So we can't really see how many times p is less than 0.05. So at the very end of the code, there's a line of code that computes the false positive rate. So here what we're going to do is from our 1,000 experiments, we're going to add up all the times that p is less than 0.05. So these are our false positives. We're going to divide that by the number of experiments, and that's going to be our false positive rate. So if we go back here, what we can see is our false positive rate is 0.048. So basically 0.05. So this is literally the p-value, right? This is how frequently we reject the null hypothesis when the null hypothesis is true. So in other words, and I'll maybe reiterate this at the end, the p-value tells you like how surprising are your results, or how likely would you expect a result to be that extreme. And this doesn't really tell you anything about how important or interesting your results are. We'll talk about how to do that shortly. Any general questions at this point? Okay, so we'll try to finish up here. Let's see if I want to cover the rest of it. Yeah, so the last point that we'll cover today, and I'm going to move over this quickly, and then because I want to make sure we have time for discussion at the end. So when we're doing statistical inference, there are two other things that we're often very interested in. One is how much data do we need, right? This is the sample size question. Now, regardless, in our first example, back to the beginning, right? So when we're trying to infer something about a population, our sample size refers to how many, individuals do we study. And there are several factors that might influence how many people we recruit for a study, how many animals we test. The problem also happens if we're interested in an individual, right? So if I'm interested in individual behavior, how many times do I need to measure that behavior so I can be confident about whether this is what I call a trait of the person, something that's consistent, a state, something that might change with time, or just noise. So let's see. Okay. So we won't do this example in MATLAB. I've just already kind of generated the results. But one of the things that I think is useful to play with to get, again, better statistical inference or intuition is in MATLAB, there's a function that you can use to compute statistical power or sample size. And it's basically called sample size power. So if you'd like to compute the size, the number of people you need in a study or the number of observations, there are a few pieces of information that you need. So first, you need to specify the type of statistical test you're going to do. This MATLAB function is fairly limited to just a few different types of statistical tests. You then input what you expect to observe if the null hypothesis is true. So this might be, the difference between two groups, or for instance, the mean and standard deviation of some measurement in your control group. You then need information about what we call the mean under the alternative. So if I'm doing an intervention, how large of a difference do I expect between my control group and my intervention group? Okay, so the difference in those means is what we refer to as an effect size. And then statistical power. So we won't have time to really get into this in detail, but this is another probability. So generally we want high probability, or high power, which means that if there is an effect, I want to be very certain that I can detect that effect. So what I do here is again do some sort of simulated example. Let's now imagine that I want to compare the vertical jump height between two groups of students. I have pilot data for one group that says their average vertical height jump is 50 plus or minus 4 centimeters. I then want to determine my sample size for a study. So, a key point here is that when you determine sample size, you have to say upfront how big of an effect you want to detect. And so this is illustrated right here. Right? So if I say a difference in height of 2 centimeters. So the key point here, at least from my perspective, is that I'm upfront dictating how big of a difference I think is interesting or important. And then in MATLAB, I'm going to compute a sample size, and it comes out to be 34 individuals. So if you're not familiar with how these things relate, specifically sample size and statistical power, that's what we're going to do on the next slide. So the key things we might want to ask, or questions we might want to answer, are, if I want higher statistical power, how does my sample size change? Or if I want to detect large versus smaller differences between my groups. So again, you can just write code to do something like a simulation here, where if I want higher statistical power, I want to be very certain that I can detect an effect. That's moving from the left to right on the x-axis. Sorry, the higher the power I would like, the bigger the sample I need. Conversely, small samples are often what we call underpowered. You can't really be confident in your ability to detect true effects. Finally, if I think about the size of the effect that I care about, you get essentially this sort of opposite relationship. So the larger the effect that I'm interested in detecting, the smaller the sample size that I need. Conversely, if I'm studying an intervention, for example, and the effects are small, then I need a lot of measurements to be able to detect that. So one of the take-home points that I would generally like to make at this point is that oftentimes we're very limited in our ability to collect large samples. And when that's true, if you have small samples, then what you would ideally like to do is to study things that are likely to have large effects. Maybe we'll have time to discuss how you might do that. All right, so what are our take-home points? Variability is what I call your best frenemy, right? You want variability when you're manipulating something and measuring the effects. However, you want to minimize these what we call extraneous sources of variability. We're going to use data visualization to understand variability and the sources of variability, and to make sure that we are not misleading ourselves or others by our descriptive or inferential statistics. The third point here is that, for at least personally, I don't feel like I really gained a good understanding of statistics until I started doing simulations. And I think this is one of the best tools you can use to understand these sometimes abstract concepts about probability. Last few points. When we think about p-values, right? P-values are not the goal, right? A p-value is essentially telling you how surprising your results are. Right? I said this earlier, right? The null hypothesis is true and p is less than 0.05. Essentially, your conclusion is like, oh, these results are surprising under that assumption. But what we're really interested in, this goes back to the beginning, where we all said we're interested in understanding the world, generating knowledge. What we really care about is if we observe an effect in our study sample, we really care about how important is that effect, right? And the challenge is that statistically significant does not mean important. The terminology is a little bit, I think, problematic because significant in everyday language often means important. That is not true when we're thinking about statistics. And so the final point is when we think about how we want to interpret our results, really the thing that I'm going to argue is most important is how large are the effects that we observe and how certain are we about these effect sizes. This is challenging for people sometimes because often we have to have some subjective assessment of how important something is, right? Like how big of a coefficient of a correlation coefficient is important, how large of a difference between groups is interesting. But this is really the core of our objective here. All right, so with that, I'll wrap up. We have a few more minutes. Please, if you haven't signed in, please use the QR code to sign in, and then please provide feedback at the link here. So we have 5 more minutes in case there are any points for discussion or questions that people might have. Thank you.
Dr. Finley, so for establishing that difference, it depends more like what's you defining your project, or you based on previous research, or what ways?
Yeah, so this question is about like how do you determine how big of an effect is interesting, right? Mm-hmm. So in the clinical world, right, people have what's called a minimally clinically important difference. So if I'm studying walking speed and I can have a change in walking speed of, let's say, 0.4 meters per second from an intervention, that might allow somebody to cross the street safely if there's a stoplight. So that's great if you can have that. For most of, I would say, what most of us do at IMSI, we don't really have that. We don't have a clinically important difference. Mm-hmm. So we have to think about, and I think Monica has a point here, right? So we have to think about, okay, what does this allow? If it's about function, does this difference allow something that we think might be important? We have to make that argument, and there's no like one way to do that. Mm-hmm. So I think this is at least how I was trained. We didn't really talk about this point in a lot of my training, but it's the key thing that we really care about. How important is this effect that we're studying? Yeah, and so there's this question about, P, right, like right above the threshold. So, I'm not gonna say what to do, but oftentimes even if P is 0.049, you should still be talking about the size of the effects, the variance in that effect across participants, because even if P is less than 0.05, maybe you have one person who had a huge effect and the others were all really small, right? Maybe on average, the effect is just really small and still not important. Similarly, you could have P equal 0.06 and you have, a lot of variability in the effect across your participants' organisms. And maybe that's important to better understand. So the major take home point is really thinking about, and I initially thought I would present about this, but there are other statistical methods you can use. For instance, reporting confidence intervals is always important. Bayesian methods are useful. So for example, you can use Bayesian models to understand the uncertainty in your estimates for a given individual as well as for the set of individuals. And there are some other, a lot of resources available on ways to reframe your results without relying solely on p-values. Other questions, comments? Do we always need p-values? Yeah, it's a hard question. So really, I mean, our use of p-values is largely determined by both journal requirements and expectations from reviewers. So some of these expectations are, like, I would say rigorous, right? Based on objective reasoning. Other expectations are based on history. So in some sense, we need them because that's what everybody is accustomed to. But I just think it's important to think beyond the p-value when you discuss your results. And we're, many of us who are here are faculty, and some of it, like, I had to change how I think about results, and it's a hard switch. I'm not going to say I've completely even done this. The other point that I didn't really have time to get into, but p-values are also largely a function of the size of your data set. So if you have a really huge data set, you almost always are going to find p less than 0.05. It just means that you can detect really small effects. Okay, so we're at 10:30 now. I'm happy to answer any other questions people may have via Slack. Again, I'm not a statistician. Statistics is a whole field to itself where a lot of these things are still debated. So if you have other resources, perspectives to share, feel free to share them on Slack as well. And thank you.